Alright, my name is Ryan Dickerson. I'm a student at the Bird School of Medicine class 2025, and this is going to be our video on the embryology of spinal cord and vertebral column development. As always, here's our mission statement for Scrubs. Scrubs is a student-driven initiative that is aiming to develop supplemental resources for current and future cohorts that will pass through Birdie. Members participate in a variety of subcommittees working to create resources for students, by students, and we're hoping that this will become a trademark of the Birdie student body and really exemplify the unique collaborative community that we offer. As always, we have a disclaimer with all of our resources indicating that our resources are made as supplemental resources and are not in any way meant to replace the Birdie faculty. With this in mind, let's go ahead and dive into the content. So in order to understand the embryology and the development of the spinal cord and the vertebral column, I think it's best to do a brief overview of the adult anatomy so we know what the end result should look like for development. So going through the general vertebral structure, it's important to remember that our vertebrae are composed of a body, the pedicles, the lamina, and then we have our spinal processes. Okay, so all of this together is referred to as the vertebral arch in red. So we have the vertebral body, the vertebral arch, and the space in the middle is going to be our vertebral foramen, which is where the spinal cord is going to sit in the adult. Right. So big things to keep in mind as we're thinking about this video: the vertebrae are going to form segmentally. Between each vertebrae, there is going to be an intervertebral disc. Between the vertebrae, the spinal roots come out through the intervertebral foramen, whereas the spinal cord will sit within the vertebrae within the vertebral foramen. So as I mentioned, it's important to note that we have the intervertebral disc that sit in between each vertebral body. And the intervertebral disc is composed of two segments. The innermost segment is composed of the nucleus pulposus, whereas the outside is composed of the annulus fibrosus. Okay, so these are two different tissue types. And we're going to see that they're going to derive from two different structures embryonically. Again, here's just a side view, just really pointing out that the vertebral bodies are going to sit anterior to the spinal cord, which is going to be encased in the vertebral arch. And this entire canal that we're making is called the vertebral canal, and that's where the spinal cord is going to sit in the adult. Okay, so with this in mind, that should give us a pretty good idea of what we should look like in the adult anatomy. Now, I do want to mention with the spinal cord organization that we have the spinal cord within the vertebral canal, and each one of these spinal nerves is coming through the intervertebral foramen. Okay, and that is at the associated spinal level. Now, in the adult, the spinal cord is going to end at the level of L1 or L2. This is known as the conus medullaris. So that is the end of the spinal cord. So at the level of L1 and L2 vertebrae. Right. All of these spinal nerves that come down distally, and this collection is known as the cauda equina. Okay, and we need to understand these terms. The cauda equina also sits within a space, and the subarachnoid space, this is going to be known as the lumbar cistern. Okay, so the lumbar cistern is this space all the way through here. Okay. Lastly, an extension of the pia mater, which is the meningeal layer that surrounds the spinal cord, is going to come all the way down and attach into the sacrum to make sure that we're anchoring the spinal cord, and this is known as the filum terminale. Right, and these are all adult structures that we should know. Okay, last thing I want to cover is just the meninges. Remember that the meninges are the layers that are covering the spinal cord. There are three layers. There is the pia mater, which is in direct association with the spinal cord. There is the arachnoid mater, which is our second layer, and then our dura mater, which is that last layer. There are a couple of specific functions of each one of these, but I want to point out the denticulate ligaments are a specific component of the pia mater that helps anchor in the spinal cord. That is the same as the filum terminale, which is again a special function of the pia mater. We're going to see why this is important later on. Right, and one more time, the lumbar cistern is in that subarachnoid space, and it spans between L2 to S2 because the end of the spinal cord, known as the conus medullaris, happens at the region of L2. All right, last thing before we get into the embryology, thinking about the actual spinal nerves and how the spinal cord is structured. Remember that the spinal cord is made up of gray matter centrally and white matter peripherally. This gray matter represents cell bodies. In the anterior aspect of the spinal cord, here we have the motor cell bodies, and these are going to go down the afferent root, or the efferent root, which are going to be efferent fibers through the ventral root going into the spinal cord, or sorry, spinal nerve, and then we have afferent fibers going through the dorsal root, which is sensory information going into the dorsal horns. So posteriorly, we have sensory information, anteriorly, we have motor information. right? So there's a pretty significant delineation between anterior and posterior within the spinal cord, and we're going to see how that develops embryonically throughout this video. So with all of that in mind, let's go ahead and dive into the development of the spinal cord and the vertebral column. So first, we need to recall that at this point in development, we have just formed our trilaminar disc. Our trilaminar disc occurred because we had the primitive groove neurostreak, and we had the gastrulation of tissues inferiorly, and replacing that hypoblast layer with endoderm. So now we have three layers. We have the endoderm layer, we have the mesoderm layer, and remember this mesoderm is specifically called intra-embryonic mesoderm, and then we have the ectoderm layer superiorly. All right, so we're going to start this video with the understanding that we've already formed the trilaminar disc, and now we're going to progress forward. So in order to form the spinal cord, and which we'll talk about all the steps along the way, the first thing that has to happen is the notochord needs to form. So the notochord is a condensation of some of the mesodermal cells that are situated in the midline of the developing embryo. So these are mesodermal cells within the midline of the embryo, and that is formed by intraembryonic mesoderm. All right, so we formed a trilaminar disc. So the mesodermal cells are going to extend cranially and caudally, and this is between the ectoderm and endoderm layers. So in this bottom image, this would be our ectoderm, whereas we have our endoderm inferiorly, and right in the middle, we have this consolidation of cells, which is going to become our notochord. All right, so there's a couple of functions to the notochord that we need to keep in mind. The notochord is going to be a rigid support. It's mesodermal cells. It's right in the midline, and it's going to help build the eventual axial skeleton. Right? It is the primary inducer of the neural tube and somite development, so it's sending signals to allow us to progress with neurulation and formation of the neural tube. Now, a couple of definitions that I want to point out. A somite is basically a building block of mesoderm that is going to be situated on either side of the neural tube when it forms, and it's going to give rise to the muscles of the axial system as well as the skeleton within the axial system. And the axial skeleton specifically refers to the skull, the vertebral columns, and the thorax. Okay, so we know that a notochord is going to form. So the notochord forms is in between the ectoderm and endoderm layers. We can actually see it better here. So this is going to be mesoderm in the middle. Right in the center of the mesoderm, we have the notochord, which is that consolidation of mesodermal tissues. Now, the overlying ectoderm is going to be induced by the notochord in a process called neurulation to become neuroectoderm. So what we can see is that with neuroectoderm, some of these cells that are lateral to the notochord are going to increase in height, so become columnar cells. And the central cells don't really change in shape. So this growth in height is going to actually cause the ectoderm to form folds. So here we can see the folds starting to form. And we're going to call these folds the neural folds. The space in between the neural folds is known as the neural groove. And this is happening right down the midline of the developing embryo. So I also want to point out, right at the top of each one of these folds, we have specialized cells, which we're going to call neural crest cells. So this is the neural crest tissue up here in dark red. Eventually, as there's increased pressure of these folds as they're forming to come together, they're going to fuse. And when they fuse, they're going to fuse, and then the ectoderm is going to go back over top. So all of this is neuroectoderm, right? And this is going to form our neural tube. So this structure here that I'm highlighting is the neural tube. You can see that those neural crest cells have now migrated. They are now underneath the ectoderm, but they're in a posterior lateral position to the neural tube. And we can see that here. So neural tube with neural crest cells in the posterior lateral position. Now, this fusion doesn't happen all at once. So what I want to point out is this fusion happens in the midline. So it starts right in the middle of the back, and then it's going to extend cranially towards the head, and it's going to extend caudally towards the rear. So the cranial end is going to close first, and that's going to happen around day 25 of development, whereas the caudal pole should close around day 27. Right? And when we have these spaces that are open before they fully closed, we're going to call those neuropores. So the neuropores close at days 25 cranially and days 27 caudally in a normal developing human. So I mentioned briefly about neural crest cell migration, but here it goes again. We have the neural folds, the tip of which we have the neural crest. When these join together, that is going to cause the neural tube to now be in between the endoderm layer and ectoderm layer. So it's in the same plane as the mesoderm, but remember this is neuroectoderm tissue. And the neural crest cells are going to invaginate and they're going to migrate into a posterior lateral position. So again, this is posterior, this is where the back is, and this will be the eventually belly. So posterior lateral position to the neural tube. And what's important about this is that the neural crest cells are going to come up multiple times throughout embryology because they're going to derive specialized structures. One of these structures is the dorsal root ganglia. So if you recall from adult anatomy, the dorsal root ganglia are attached to the spinal cord and they are located in a dorsolateral position because they're dorsal root ganglia. So it's in the same position that we see here with these, the developing of the embryo. The other thing that we're going to have from neural crest cells that are going to derive is the peripheral ganglia, like peripheral nervous cell bodies, as well as the support cells or the glial cells of the peripheral nervous system. I also want to point out, and we'll cover this later, but they form two meningeal layers. And the specific meningeal layers that the neural crest cells are going to form are going to be the pia mater and arachnoid mater. So the two innermost layers, but not the dura. Okay, so pia mater, arachnoid mater, but not the dura. So we've now got this neural tube that's formed right in the middle, just posterior to our notochord. Well, we know what the eventual structure should look like. We should get the ventral horns and dorsal horns with the gray and white matter. So during development, we have the neural pores close, which is going to form a nice tube all the way through. And then in the lateral regions, we're going to get indentations, which are going to be called the sulcus limitans. And the sulcus limitans on either side are going to separate the neural tube into an anterior region and a posterior region. The anterior region is going to be called the basal plate. Okay, so the basal plate is anterior. The posterior region is going to be called the alar plate. And again, this is posterior alar plate. The basal plate is going to eventually develop into the ventral horns of the spinal cord, whereas the alar plate will eventually develop into the dorsal horns. And you can see here, this central canal is going to get much smaller in the adult derivatives. But this is, remember, sulcus limitans dividing the basal plate and the alar plate. Now remember, because we understand the functions of the adult structures, the alar plate is deriving everything posteriorly, so this means it's going to be developing the components that are receiving sensory information and giving rise to the dorsal horns. Because we have our dorsal root ganglia, which is developed from our neural crest cells, giving information, that sensory information, into the dorsal horns. Whereas the basal plate is primarily going to be motor in function, so it's going to derive the ventral horns, and that is the efferent fibers that are going to be sending out through the ventral root into the spinal columns or spinal nerves. All right, so that is how we get that separation between motor function and sensory function with spinal cord development. So that's great. We have the spinal cord developing, but remember that in the adult, the spinal cord is surrounded by the vertebrae. So that means that the vertebrae also have to develop around the same time. And the way this happens is we talked about the notochord being located centrally, made out of mesodermal tissue, is inducing neurulation of the ectoderm, forming neuroectoderm, which is eventually going to form the neural tube. Well, while this is happening, the mesoderm that's in this region is going to divide into three different regions. First, we're going to have the paraxial mesoderm. The paraxial mesoderm is directly beside the notochord. Right? Beside that, we have the intermediate mesoderm. And then most laterally, we're going to have the lateral plate mesoderm. For the remainder of this video, we're primarily going to be focused on this paraxial layer. The intermediate and lateral plate will come up in later videos. So again, the mesoderm, this is intraembryonic mesoderm, that is directly beside the notochord, is going to differentiate into paraxial mesoderm. Right? And eventually, what will occur is these will separate into paired somites, one on either side. And remember, those somites are building blocks. Basically, think about them at each vertebral level. And that means that they're going to receive segmental innervation and blood supply. So again, building blocks that go up along the spine. So these somites that are forming just on either side of the paraxial mesoderm. So this is the paraxial mesoderm differentiating the somites. The somites are going to differentiate once again into two separate components. You're going to get the scleroderm, which is going to be this tan color that is anterior medially, and the dermomyotomes, which are going to be located posterior laterally. So over time, these structures are going to give rise to the axial skeleton, and associated musculature. So the scleroderms, this is going to derive the spinal column, whereas the dermomyotome is giving rise to the musculature and the associated dermis. So you can see here, if we look at the sclerotome, that's this 10 component, it's going to eventually come and surround the notochord. And it's going to surround the notochord. We'll talk about this in the next slide. It's called the centrome, and that's going to form your eventual vertebral body. And remember, in the adult, we have the vertebral body located anteriorly, and the vertebral arch, inside of which we have the vertebral foramen, which is where the spinal cord sits, which is what we see here. So this is how these are developing. Now, that said, you still need your dermomyotome because you have muscles associated with the vertebral column, and that's exactly what this is giving rise to. So you can see those in blue here. All right, so this is the differentiation of the somites, which are made from paraxial mesoderm, that then differentiate once again into sclerotomes and dermomyotomes that give rise to the spinal column and ribs, that's the sclerotome, and then the axial musculature and associated dermis, which is the dermomyotomes. So going into a little bit more of the differentiation of the sclerotome, remember that this is going to be forming the vertebral column. And the way that this happens is the sclerotome, which we see in tan here, that's one of those components of the paraxial mesoderm, it is going to be surrounding the notochord. So when the sclerotome surrounds the notochord, that region we refer to as the centrum, which you can see written here. So the centrum is going to be the location of the future vertebral bodies. And so the vertebral bodies are going to be forming around the notochord. And we know that the vertebral bodies in the adult are going to be located anteriorly to the spinal cord, which is developing by the neural tube just posteriorly to the notochord. So this makes sense. And what I want to point out is that you have the sclerotome from either side contributing to the vertebral body. And there's going to be two processes that occur on either side. That's called endochondral calcification. That's the specific type of bone formation that we have here. And there needs to be a ossification center on either side of the vertebral body, so one on either side. And we'll talk about why that's important in the clinical anatomy correlations. So the other thing that I want to point out is when you're thinking about the vertebral column in the adult, it's not just the vertebrae. You also have the intervertebral disc that sit in between each vertebrae. And if you remember from adult anatomy, there are two components to the intervertebral disc. You have the nucleus pulposus more centrally, and you have the annulus fibrosus located more laterally. So in forming the intervertebral disc, what happens is you have mesenchymal cells, which are mesoderm, that enter embryonic mesoderm, and it is invading in between the forming vertebral bodies to form the intervertebral disc. And remember, the vertebral bodies are forming right along the notochord, which you can kind of see here in green. So as these mesenchymal cells invade, they're going to form the outside of your intervertebral disc, known as the annulus fibrosus. So there's mesenchymal cells forming the annulus fibrosus. The notochord in adults is going to persist, and it's going to give rise to the nucleus pulposus, or that central component of the intervertebral disc. So in summary, you have the sclerotomes, which are coming in as paraxial mesoderm, surrounding the notochord to form the vertebral bodies, which will eventually form the vertebral arches and the entire vertebrae. But then in between each one of those vertebrae, remember because we have segmented somites, we have invasion of mesenchymal cells forming the intervertebral disc. The more lateral components, known as the annulus fibrosus, are formed by that intraembryonic mesoderm, which in here we're just calling it mesenchymal cells, which is connective tissue. And then centrally we have the notochord, which is going to persist as the nucleus pulposus. And that's going to give you your rise to your vertebral column with both the vertebrae and the intervertebral disc. So when we initially start development, this is happening directly anterior and posterior to one another. You have the L1 vertebrae corresponding directly with the L1 spinal segment. But as you grow in development, the spinal cord grows much slower than the vertebrae does. So the vertebrae are going to start growing faster and faster and faster, and these spinal roots that are going through the intervertebral foramen are going to be locked in place. So as the spinal cord continues to grow, as the vertebral column continues to grow inferiorly, the spinal cord is locked into place, and it starts to stretch out these spinal roots. And eventually, that's going to lead to what we know as the adult, the conus medullaris, the spinal cord ending at the level of L1, the lumbar cistern, which is the space between L1 and S2, as well as the cauda equina, which are these really stretched out spinal roots that are going to their associated intervertebral foramen. Right. So that is due to differential growth of the vertebral column compared to the spinal cord. Right. And you can see that in summary here, the vertebral column and dura is going to outpace the spinal cord in terms of growth. The conus medullaris is going to seemingly rise. So it initially was down at the C1 level. It's going to come up to the L1 level in the adult. And in newborns, that's usually around the level of L3. And now you have formed your lumbar cistern, filum terminale, and cauda equina. Additionally, when we're thinking about the developing nervous system, we need to recognize that the meningeal development has to occur as well. And again, the meninges are the covering for the central nervous system. And it's broken down into 3 components. You have the pia mater, which is in direct association with the spinal cord. You have the arachnoid mater, which is your second layer. And lastly, your dura mater. So, initially, around the neural tube, which is going to be forming your spinal cord, you're going to have mesenchymal cells surround the neural tube and form what is called the primordial meninx. So the primordial meninx is going to differentiate. You're going to have the external region, which mesenchymal tissue has invaded, form the dura mater. But on the internal surface, near the spinal cord, you're going to have neural crest cells invade. So the neural crest cells invade, and they're going to form what is known as the leptomeninges. And the leptomeninges are referring to the pia mater and the arachnoid mater. So those 2 innermost layers of the meninges. Okay, so one more time, we have the primordial meninx that is made up of mesenchymal tissue. The external components are going to give rise to the dura mater. Internally, closer to the spinal cord, you have invasion of neural crest cells, which is going to give rise to the leptomeninges, which refer to the pia mater and the arachnoid mater. So, in summary of the developing of the spinal cord and vertebrae, the general steps are that you have the notochord forming, which induces the formation of the neural plate. That leads to the neural folds, the neural grooves, and the neural crest cell to form. When the neural crest fuse together, the neural tube is then going to invaginate inferior to the surface ectoderm, and the neural crest cells are going to migrate to their appropriate location, which is going to be posterior lateral to the neural tube. Initially, the neural tube is going to start closing along the midline of the back, and it's going to extend in a cranial and caudal direction until it will eventually close the neuropores, which again are the openings before it is fully closed. At this point, we have the neural tube, which is going to develop into the spinal cord, and that is going to form based on the basal plates and alar plates of the neural tube. And remember that those are divided by the sulcus limitans along the lateral margins. Additionally, we have to form our vertebral column at the same time. So we're going to have the paraxial mesoderm form somites on either side of the notochord. These somites are going to differentiate into sclerotomes and surround the notochord and spinal cord, and that is going to eventually give rise to our vertebral column, right? And then we're going to have mesenchymal cells invade in between the vertebral bodies, and this is going to form our intervertebral disc. Remember that the notochord is the nucleus pulposus of the intervertebral disc in the adult. All right, and again, we have differential growth rates of tissues, meaning that in the adult, the end of the spinal cord is going to be higher than the end of the vertebral column. And as just a general overview, here's a table of the different germ cell layers and their derivatives. So we have the neural tube, which is made up of neuroectoderm, which is giving rise to the brain and the spinal cord. The neural crest cells, which are also neuroectoderm, are giving rise to the dorsal root ganglia, the peripheral nervous system glial cells, the pia mater, and the arachnoid mater. The paraxial mesoderm, which is the mesoderm directly beside the notochord, initially gives rise to somites, which divide one more time into dermomyotomes and sclerotomes. The dermomyotomes are giving rise to the axial musculature and the dermis of the skin, whereas the sclerotomes are giving rise to the vertebral columns, the ribs, the dura mater, and the annulus fibrosus. And then lastly, we have the notochord, which is initially formed by intraembryonic mesoderm, and it is going to give rise to the nucleus pulposus. And again, remember that the notochord was very important in this process because it is the it is the structure that is going to induce signaling to cause neurulation and formation of the vertebral column to occur. All right, so with that understanding, we're going to talk briefly about a couple of clinical anatomy points. First, neural tube defects. So neural tube defects can happen anywhere along the spinal cord. We're primarily going to be concerned with them happening at the caudal pole near the tailbone, and this is going to be called spina bifida. More anteriorly, if it happens in the region of the skull, this can be anencephaly, meaning lack of a skull, and you have different components that can happen along the length of the spine as well. But what we want to point out is when you detect these in development, usually this is done via a test for alpha-fetoprotein. So if alpha-fetoprotein is elevated, it raises concern for a neural tube defect. So make sure to remember alpha-fetoprotein associated with neural tube defects. And over time, what we've learned is that most of these defects are due to inadequate folate intake during early development, primarily within the first 2 months of development of the embryo, because that is when you're forming the vertebral bodies, vertebral arches, and the neural tube. All right, so that is the main association with neural tube defects is going to be folate deficiency. So talking specifically about some of these neural tube defects, we're going to focus on the lumbar region, and we're going to talk about spina bifida. So spina bifida has a couple of different flavors. First, we have spina bifida occulta. So this is due to failure of the vertebral arch to form. So remember, this is mesodermal cells. And you don't have any invagination of the meninges or the spinal cord. All right, so it's just failure of the vertebral arch to form. Oftentimes, this will have no clinical symptoms, but you will hear the telltale tuft of hair in the lower lumbar region is usually associated with development of spina bifida occulta. Next, you have spina bifida with meningeal. So meningeal, there's a cyst with the meninges involved. So you have failure of the vertebral arch to form, same as you do in spina bifida occulta, but now you have the meninges invaginating through, but not the nerves. Okay, so this is just the meninges associated with the cyst. Lastly, you have myelomeningeal. So same as meningeal, but now you add myelo. So this is going to include the spinal nerves. So you have the vertebral arch defect plus the meninges plus the spinal roots. And this is going to lead to spinal nerve defects below this region. Now, some additional terms that you might see spina bifida with myeloschisis. This is actual failure of the neural plate to close, and this is spina bifida in its most severe form, and it's always associated with nervous defects. If this expands from multiple spinal segments, we'll call that rhasischisis. And so that's just myeloschisis spanning multiple spinal segments. And that's kind of a brief overview of spina bifida. Last things I want to discuss is the development of a hemivertebra or chordomas. So a hemivertebra is, you kind of see it here, this is when only one half of your vertebral body forms. And if we think back to the formation of the vertebrae and the vertebral bodies, remember this was the sclerotomes that were invaginating around the notochord. And I mentioned that there was a ossification center on either side. There's one on the right and there's one on the left. If the ossification center on one side doesn't occur, then you're going to get only half of the vertebrae forming. And when this occurs, it's going to cause significant deviation of the spine laterally, and we're going to call this scoliosis. All right, so scoliosis could be caused by hemivertebra formation. Lastly, chordomas. Chordomas refer to a tumor of the primitive notochord derivatives. So chordomas can happen anywhere along the location of the notochord or the associated structures in adults. A lot of times these will happen at the base of the skull, known as the clivus specifically, or the sacral region, but you can also have them all the way along the spine. But really, all we're thinking here is a chordoma is a tumor of the primitive notochord derivatives. All right, and with that, that'll bring us to an end of this video.